Okay, I think we're going to get started. Welcome, everyone, to the 2022 Neuropathology session of this Fall ADRC meeting. My name is Andrew Teich. I'm a Neuropathology Core Leader at Columbia, and I'm leading the session this year. We've got a fantastic schedule of speakers, beginning some updates on digital pathology working group in the Neurobiobank, followed by a rapid round of talks from junior faculty. At Columbia, we refer to junior faculty. I've told early stage investigators, and then we have two DEI themed talks at the end. And so one point, the schedule it mentioned that I'm giving the survey update. So many of you have gotten requests to fill out a survey. I think everybody's filled it out at this point, which is much appreciated. And we have those results. This was done in coordination with the Digital Pathology Working Group. And so when Brittany Duggar comes up and talks, she's going to give you an update on the survey results. So my slide? Yes. So I only have a couple of additional things to mention. Sorry. So first off, I want to thank Dr. Ann McKee, who has been on the committee for 3 years and is now rotating off. And welcome Dr. Brittany Duggar, who is joining us. A couple of updates from NIA that Nina wanted me to update you on. So I know that recruiting people for brain donation is sort of an ongoing challenge for a lot of centers, and so there's additional resources that are now available in terms of literature and sort of websites for people to visit to encourage brain donation. And so you can make yourselves available, avail yourself of those resources, and Nina has more details on that. And then I also wanted to update people on the late meeting, which is coming up. So if you're interested in that and haven't signed up yet, please do so. And then the only other thing, wow, I'm almost at 10 minutes already, so I will quickly get through this. Mark, VCID, I'm just alerting to you about this particular group. So this is a group, Drs. Julie Schneider and Matthew Frosch are part of this working group. There's several centers around the country you can see there on the map, and the goal here is to come up with biomarkers for vascular disease. And I will skip right to the chase. They're on their second iteration, and right now there's an ongoing conversation about the best way to collect pathology data, documenting vascular pathology and how to incorporate that into their biomarker work. Here's what we currently collect for the NAC form. And so some questions that group has right now is, what's the best way to continue to collect this data and to sort of facilitate the general goal of documenting vascular disease and fold it into sort of the goals of this particular working group. So I've got a lot of information here on the slide. Basically, you can sort of read through this. If you have any questions, you can talk to me, or Julie Schneider is the one who is leading this effort, and her email, I think most of you know her, but her email's there at the bottom. Specifically for the group, one reason I wanted to bring this up is there's some discussion about what's the best way to capture vascular pathology as you're doing your neuropath sign-outs, and should we, reformulate in some way the way we're currently collecting it on the NAC forms. Obviously, we don't want to greatly increase the burden of work for people, so is there a way to, rather than just add to it, reformulate it in a way that sort of is more updated with the current state of understanding of what's relevant vascular-wise, or should there be some optional module? So I just wanted to alert you that that's ongoing, and Julie Schneider can answer questions about that. And so that's all I have. I'll get out of the way here. There's a number of great speakers we have, and I will just introduce the first one, Dr. Brittany Duggar. I think you all know her. She's at UC Davis. She's the Neuropathology Core Leader there and also runs her own lab. She's an Associate Professor of Pathology and Laboratory Medicine, and she will be giving you an update on the Digital Pathology Working Group.
Thank you, Dr. Teich, and I've been told not to touch the mic. So again, I'm Dr. Brittany Duggar, just so I can identify myself after not so much coffee today. Digital Pathology Working Group, just to give you a brief history and a big shout out to Dr. Nina Silverberg, who is a huge catalyst in forming this, as well as many other things that she does. You know, I think you got a couple Olympic medals somewhere, right? Okay. So in the spring of 2019, emails were sent to neuropathology core leaders soliciting for interested personnel, and then we got right to it in June of 2019. And we've been pretty much meeting monthly since then. We meet on the second Monday of every month from 8 to 8:45, so a great way to start the week if anyone wants to join. We have wonderful conversations revolving around digital pathology. We've had attendance from people across many institutions and centers, and the minute you start putting up logos, you're probably going to forget somebody. So I tried to capture this, but it's so great because people have taken time. Think about how busy everyone's day is, and people are taking time out of their day to attend these digital meetings, which has just been phenomenal. We've even started to get people from across the pond, I guess is the phrase, so from Europe that are wanting to sit in. So you just think of that time difference as well, which has been great. So the goals of the group, because it's always good to have goals, right? We learned that in the first sessions this morning. So we wanted to assess the needs and potential usage of digital neuropathology within ADRCs. We wanted to evaluate the feasibility of implementation of these technologies across centers and develop recommendations for their usage. Pretty simple. And so what is milestones without metrics? Metrics are always good things. So we have some metrics here. We had a 2019 survey, which many of you filled out, and I'll present some of those works as well on benchmarks. So you can't really make changes unless you know the landscape. Digital pathology webinar series, so I'll talk about that, and that was a great collaboration with NAC. So not only do they throw a great fall meeting and spring meeting, they also help out with numerous other projects. We're doing a pilot study for digital slide sharing, I'll go over that, as well as a 2022 survey, which I think that was the most recent one. And again, thank you all, because even though I'm up here talking about all this stuff, you guys submitted the data, so thank you so much, you people, inclusive language. So with the first data that we did, that 2019 survey, we actually did a guide to digital slide scanners, and this is just a very short FAQ sheet, and you can see some of the topics here that we go over, like, okay, how can I afford this? What do I do? Where's the infrastructure? And although we have a great working group, it's always great to involve people that are early in their careers. So I have great photos at the bottom of Yama Hamsafar, who is now an EMT. He worked in my laboratory, and I have my current postdoc, Dr. Rebecca Scalco, who's been helping with this. We actually turned this into a paper, and it's currently under review, so hopefully that can get in press soon. But you can see that there's a link just for the white paper if anyone's interested in that. Some of these responses, just to highlight some of the things, because these are common questions we've been getting. Okay, what scanners are people using? The most common is Aperio Leica, the Aperio Leica scanner. Most of the respondents, which is 81%, stated that they had access to a scanner. And this was really awesome. We had over a 90% response rate with the ADRCs with this survey. So really, really awesome. So again, thank you all for your responses on this. And the most common brand, Aperio Leica. Other things that I think are really important is funding. So yeah, we could go to the NIA for stuff, but it's also good to have strategic partnerships. So over 50% of the respondents stated that they had institutional support when they had their equipment purchases. And you can see some of the other funding sources, philanthropy, and S10 large equipment grants. So just to give you some ideas if you're interested in the technology. Other just short highlights. 63%, and this was back in 2019, just to date that, had scanned in less than 10% of their current slide inventory. 50% were unsure of total storage space, so those in the data statistics core try to think about that. If this is coming down the line to storage, each one of these files can be quite large, and you have multiple files per case. 53% would find the option of a centralized scanner service useful, and there's actually a current RFA out for a U-style grant to help with this. Next statistic, 47% did not have any of their ADRC's budgets allocated to digital path, so again, metrics. And then 50% noted that they shared digital files. Other things to highlight from the working group, and big shout outs again to NAC with Hannah, and then Dr. Maggie Flanagan at Northwestern. They've been instrumental in organizing this webinar series. So we are on YouTube, and I don't have the exact statistics, but I've been told that we're more popular than select videos of cats scared by cucumbers. We do have a lot of topics that we went over, so feel free to visit us on YouTube, but don't Google my name, that you don't want to see that stuff. We do have upcoming webinar, and this is going to be November 14th. More information coming soon, and we're going to focus on the digital pathology infrastructure and informatics. So if you know of anybody that's interested, feel free to share, right? It's trying to decrease those barriers. Then when we move on to our pilot study, and there's a lot of awesome people. So even though we have the big digital pathology working group, we kind of spun off and have this small tiger team. And again, you can see there's a lot of names on that from a lot of different institutions. And the biggest thing is, can we drive the road? So can we try to understand the workflow for having a centralized database to identify those pain points? And so we're using what is called the Digital Slide Archive. This was initially set up at Emory and from the National Cancer Institute. So there was a huge bolus of funding that went into that to build that, and then we're trying to capitalize on that infrastructure. And so here's just, it's still under construction, right? It's always good to just see some metrics. So we've had a lot of different centers submit slides to this, and so you can see some of the data, and then we're having it to where you can search it. So, how many H&E slides of, this type of area for this case? So we're having all those metrics searchable within this. But again, it's still under construction. And this also led to the second survey that we did. So again, thank everybody for filling this out. And this started in the summer of 2020. And this kind of also paired along with, there's a lot of conversations on the national level about harmonization. But how do you harmonize if you might not understand the metrics of where you're at? So this is a tough thing because a lot of us have brain banks that have been run for many, many years. And we have certain sampling parameters that will fit our disease-specific entity for that. But on top of that, we have a lot of new ADRCs that have come on board, which is so exciting to see. So when you think about that map and all the color that's come into that map over the past couple of years, it's so exciting. So that's why we thought that this was such a great thing to go forward. And we paired with the Neuropath Steering Committee, and specific thank you to Dr. Andy Tyke, who helped with this as well. And then again, pulling in students and early career persons. So you see all those photos in the bottom corner. So two of persons in my lab, soon to be hopefully, Dr. Kevin Nezuku, who just got into medical school, and a former postdoc of mine, Catherine Luco, aided, as well as a graduate student at Emory. So they all tried to help build this survey, try to understand all the feedback. And basically, we just wanted to see what people are doing. How are you brain banking? What are you sampling? What are you staining? And so with this, here's some of the data. And again, thank you for the response rate. We had a 95% response rate. And this not only includes the ADRCs, but we also had people from other brain banks wanting to do our survey, which is huge. I was just so excited for that, as well as the group was. So with amyloid beta antibodies, and this is just the top 5 responses, most people are using 4G8. For the tau antibody. And again, I have all these data, so feel free to email me if you want to see them. The top antibody was AT8 for the tau, but you can see there's some other ones. And then for alpha-synuclein and TDP, we kept with the language that's very similar with the NAC forms. So with alpha-synuclein, it's kind of split 50-50, who's using the phosphorylated versus non-phosphorylated, but TDP, 43, majority of people are using phosphorylated. And then this, I know this is a lot of numbers right after lunch, but hopefully you can just appreciate the heat map here. And again, thinking of this whole concept of when you get brain tissue or you get data from certain institutions that we all sample similarly, but there's also differences. So on this chart, hopefully you can appreciate on the one column, these are all different areas. So you think all those different areas of the brain and what stains are done on that area. And so a lot of this parallels what was put forth in the NIAA criteria, right? But you can see that there's a lot of other things that are creeping in, like olfactory bulbs. Some people sample, but not a lot of people. So just very interesting data as we start to talk about, okay, we're bringing a lot of centers online. How do you want to sample your tissue? How do you want to stain your tissue? What resources do you have available to accomplish that? And so that's about it. I have 11 seconds left. So we do love to talk and collaborate and have conversations. So if anyone's interested about the webinar series, and I did get permission from Dr. Flanagan, so you can email her. I have permission for myself for the Digital Pathology Working Group. And then if you're interested in contributing slides to the pilot project, you can email Dr. David Gutman. And so with that, I can take any questions. On behalf of the working group.
Thanks. We have 5 minutes for questions. If you have a question in the audience, come up to the mic.
Or I could show a video of a cat with a cucumber.
Fantastic. Great work, as always, from the Neuropathology Cores and the steering committee. I would just like to encourage you, especially in the theme of the entire conference on diversity, equity, inclusion, for your pilot project, I think there should be a specific reach out to centers, not just to contribute, because those were very low numbers of cases, but let's make sure that the digital pathology methodology is working. I'm sure that it will, but I think we need to prove it to everybody in the world that this works for a diverse, inclusive cohort that we're following at all of our centers.
Yep, we're already ahead of you. So that was one of the big, I didn't put that. So thank you for announcing that, Kajas.
Hi, great talk. Jeff Nerschel, Stanford ADRC. What are the range of whole slide image file formats that you've been getting, or you've been scanning them all on site? And how is the ADRC Digital Pathology Working Group working with the Digital Pathology Association for sort of coming up with standardized file formats?
That's a great question. So all of that is kind of associated. So we don't have DICOMs, yay. Basically, all those different scanner types that I showed, each one of those has typically a proprietary file. So of the majority of the files are SVS files, but there's CZI files, there's big TIFF files. So you're point on in talking about the diversity. So we have worked with the Digital Pathology Association in the fact that Eric Glassy, who's one of the members, has actually come to UC Davis, so him and I have had conversations. I really think that that's a good idea, that we could bring him more into the fold in the Digital Pathology Association, into our meetings. So that's a great point. I know we've had like the hallway conversations, but having something a little bit more in alignment, so thank you.
Hi, Tom Beach from Arizona. You know, we're all often bedazzled by new technology, and we might just jump into it at huge cost without really figuring out what's it going to cost us in terms of effort, and what's the benefit-risk ratio, and is there anything else we could be doing with a huge amount of money? And I'm a little worried about this because it's going to require neuropathology core budgets to go up, because just purchasing and running these machines and paying for the storage is one thing, but there's a lot of effort behind it. That's going to be effort for databasing, statistics, and for the neuropathologists to run it. I see maybe another up to 50% increase in a neuropathology.
Can you add a 20% to that too?
Yeah, 70% then. Okay.
There's inflation. Yeah. Or wait, do we call it edema or do we call it inflation?
All right, thank you so much, Brittany.
You're welcome.
I am going to, we're a little bit ahead of schedule, which is fantastic. I'm going to, is this working? Yeah. I'm going to next introduce our Dr. Dirk Keen. I'm not facing you guys. So Dr. Keen is a associate professor at University of Washington in the Department of Pathology, an adjunct associate professor of ophthalmology and neurological sciences. He is director of the University of Washington Medicine Biorepository and Integrated Research Laboratory, and he's going to be giving us an update on Neurobiobank, and he is doing that remotely. So, Dr. Keen, I hope you are here.
I'm here. Can you hear me? Yeah, so we should be sharing my slides, and there we go. Hey, everybody, thanks, and thanks, Brittany. You are always so much fun to go after, so I'll do my best. Today, I'm going to summarize progress that we've made with our working group focused on trying to find opportunities to collaborate and bridge neuropathology cores in the Alzheimer's centers and the NIH Neurobiobank. I've presented on this several times in the past, and this is really an update of our progress. I'll go through some of this pretty quickly, but just to remind everybody, there are 37, I believe it's 37 Alzheimer's centers, all of which, well, there's 33 that have neuropath cores, and then the exploratory centers, of course, are developing their neuropath cores. And there are 6 Neurobiobank sites across the country. And let me just try to put on a laser pointer here. The Neurobiobanks process a lot of donor brains across the spectrum of normal adult and pediatric brains, as well as diverse injuries and diseases, and have been overwhelmed over the last few years, frankly, with donors with Alzheimer's disease that wanted to contribute. When you combine that with the 80 Centers panel recommendations that came out back in, boy, it's been 4 or 5 years now, where there was great emphasis on several different aspects of improving access to research. And I think as the theme for this meeting is really pronounced, it's really important to emphasize how difficult it has been in many cases in neuropathology to offer brain donation across the spectrum of underrepresented groups and different ethnic groups, et cetera. And something that we're all working on, and hopefully this pilot will help identify additional ways that we can maximize that. But just to highlight, the panels recognized that we needed to improve our research opportunities when it comes to neuropathology, both with very well characterized research participants, with control donors, so cognitively normal donors, and to develop guidelines and harmonized approaches so that we could better share data with each other and support researchers that are using these resources. Combine that with recommendations from the research subcommittee for the advisory council, which specifically called out the Neurobiobank and the ADRCs to try to build collaborations to support access to brain tissue for researchers and more critical brain tissue resources in our banks. And so for that, the working group was formed back in 2018. It's been a real pleasure to work with this amazing group. There are many more, but these are the sort of key elements. And Nina has really been the driver for a lot of this. She's really been supportive of increasing access to brain donation across diverse communities and making sure that researchers have access to those brain tissues and that we find a way to work together, and has been really driving this. And I also want to call out Abigail Soyambo and Daniel Miller, who are in charge of the Neurobiobank and have been instrumental in helping the collaboration. And then finally, the Neurobiobank directors, Dr. Beretta, Haratunian, and Scott, have just been phenomenal. So with them and Dr. Huttner at Yale, Neuropath Core, and then of course, Dr. Kofler at Pittsburgh, we've been working on a way to explore opportunities to improve neuropathology research in the context of ADRD. And so we've been having these monthly meetings and developed a pilot. And our goals for our pilot were pretty diverse. We wanted to develop communication and collaboration pipelines between the donors, the coordinating agencies or coordinators, and the actual brain banks. We needed to build communication strategies to reach out to donors and to communicate with people that the researchers that want to use these tissues. We had to develop standards and guidelines as far as how do we prioritize potential donors and inclusion and exclusion criteria that will work not just for our individual sites, but across our collaboration. We had to build protocols that included geographical limitations for the AD centers, trying to determine how best these centers could participate and what our site-specific areas of interest were, and then to really test run this to find out how it's working, to improve it, and then hopefully to build a model or identify a model that might be useful to the broader ADRC community. And so with that, we joined Tish Hevel. I don't have her picture here. She's amazing. She started the Brain Donor Project, which is the national referral agency for the Neurobiobank now. And so we work with Tish when she has a donor that comes to her in our geographical area and fits the criteria that I'll talk about in a minute. She refers them directly to either Pitt or UW or Yale. And so prior to that, the Neurobiobank had been trying to address the issues that they've been having with having a flood of potential Alzheimer's disease donors. And so they developed this algorithm to try to determine who to accept. And so if the donor was already registered with Neurobiobank, they would of course accept them. If they were an early onset AD, they would accept them as a high priority. Anybody from an underrepresented background or minoritized population or community would be accepted, trying to emphasize the opportunity to add diversity to the brain biorepository resources across the nation. If the donor didn't fit any of those criteria and wasn't diagnosed by a physician, then they were declined. If they were diagnosed by a physician, then they were progressed through to either, if they have family history, a significant family history and possible genetic driver or risk variants that they might have, they would be accepted by Neurobiobank. Or if they'd been in a clinical trial, they would be accepted by Neurobiobank. And so for all the people that were diagnosed by a doctor, but didn't fit any of these other categories, they were declined. They were being declined. And those are the people that we decided would be referred to, and I'm trying to forward my slides, that the Alzheimer's centers could support, at least initially. And I know for many of you, this is, we're interested in these Neurobiobank referred groups as well. Any kind of the Lewy body dementias, the FTDs, et cetera, are of great value to the Neuropath cores, as well as obviously any of the other categories, like people that are from underrepresented backgrounds, people with family histories and genetics. And so those are still referred to the Neurobiobank as prioritized to them, but the ADRCs have been getting the AD donors in our geographical area, and we were able to set whatever we wanted. We decided on 100 miles for either people with AD dementia or controls, and we also accept people with clinical MCI, vascular dementia, or some other dementia that's not otherwise specified. And so that's how the program has been working. The workflow is once the Brain Donor Project identifies someone for one of our centers, they refer that person to us, and then they go into our normal workflow where we reach out to consent. So either Julia or myself or our teams. And then once they die, they go straight into our normal ADRC Neuropath Core workflow. So we're basically adding brains along the very same pipelines that we would be using for our clinical cores, and having tissues that then should align with those other brain resources, but that are affiliated with the Neurobiobank. We are trying to harmonize with Neurobiobank because these people will be jointly they'll be affiliated. And so unlike some cores, we're getting serology on our donors. We're getting toxicology testing through a pipeline called NMS, a company called NMS. And we're trying to collect basic data elements, required data elements for Neurobiobank that we all collect anyway as part of our Neuropath course. And so we've made great progress in this pilot. The things that we're still struggling with is developing a follow-up approach like the Neurobiobank has for interviewing families to collect the information that will make these donors much, much more valuable scientifically. And then, of course, we want to ultimately also upload these donors to NAC as part of the brief data set. And so our progress to date, this is Pitt and UW. So Yale's had some challenges with the pandemic in getting up and running. But Anita is raring to go. But so far, it's just been UW and Pitt. We've had 90 people consented through this program. You can see the predominance of females. You can see that we have quite a lot of controls in here, and the age range is pretty vast. From a diagnostic perspective, a clinical diagnosis, we've got about a third to a half have some form of clinical dementia or diagnosis. 38 people that registered with us have died and come to autopsy. Again, there's a female predominance. Almost all of these people are demented, as you would expect. That the AD, the people with AD, are the ones that are dying and donating their brains. I would argue that, except for maybe a Lewy body and the PD cases, all of these donors would have been rejected by Neurobiobank. So these are people and families who've been offered the opportunity to donate their brains, who probably wouldn't have if they were going through the Neurobiobank selection criterion. This is just a little bit more data on the cases that have final neuropath. 28 of them are finalized, and you can see we have a high proportion of high ADNC. We do have a wide variety of other pathologies, including a good mixture of Lewy body disease and a pretty good mixture of late. And so I think these brains are going to be highly valuable for research, especially research that doesn't require really in-depth longitudinal clinical or psychometric data. So going back to our goals, we've achieved almost all our goals for this pilot. We still have to, we really need to find areas where we can improve. And I think, having a better dispersal of the different diagnoses to the Alzheimer's centers and the Neurobiobank is something I've been lobbying for from day one. I think we would all appreciate the opportunity if someone with FTD in our community wants to donate, that we would have the opportunity to do that. And so we're working on that. And then building models to extend this to the broader community, I think, is something that is our ultimate goal, making this palatable, not just palatable, but valuable, not just to the neuropath cores, but to the communities where there are people that will be able to get a neuropathology diagnosis, and to local researchers and national researchers who can find these donors either locally or nationally. And that comes to the last little bit, which is our tissue sharing protocol. We've designed this as affiliate sites to the Neurobiobank. We have our cases listed in the Neurobiobank catalog, and people can come to us through the Neurobiobank website, which would refer to us, or can come through our normal workflow. If we use Neurobiobank tissues to fill tissue requests, we would then report back to Neurobiobank on a quarterly basis that the progress that we've been making. And there may be opportunities down the road to coordinate with Neurobiobank if they're getting certain requests. And then tissue sharing would be through the normal workflow. I think that's going to work pretty well if we're able to get that set up and running, but we haven't done any tissue sharing yet, actually. So the benefits of the program, I think we've definitely developed a really good collaboration, and I've gained a lot of new professional colleagues who I have tremendous respect for. I think we've been able to enhance brain donation for our universities in Pittsburgh and in Seattle. I think we've been able to learn a lot about how other people do things, for instance, toxicology screening. We have methods that are continuing to be updated. And I think we really have the opportunity locally to maximize the impact by using the expertise and the pipelines that we have to take these donations, rather than having somebody far away trying to coordinate that. So that's where we're at with the pilot. I'm sure I'm running long, but I have 3 more slides. Nina really wanted me to update people just on the progress that we've been making on the TBI related neuropathology common data elements. So this is a collaboration with a number of different people. Dr. Crary, Folkerth, and I co-chaired this working group to try to develop the neuropathology CDEs for TBI, particularly chronic TBI. It's been a really great collaboration with a lot of amazing people at the NIH and with the data submission team. And just real briefly, and thank you, Dr. Crary, for this nice slide. The approach that we took was our usual approach, which is we get a brain and we sample it, and then we have a microscopic, but to try to make it much more open and available for different techniques, so that we're not constrained by one set of guidelines for how we sample these tissues and study these tissues, developing minimal and core classifications, but also exploratory classifications. And we really tried to make this sampling scheme amenable to being linked back into common coordinate frameworks and atlases so that we could get a better sense of the broad anatomy that was being sampled for the different centers, giving centers essentially an open-ended opportunity to develop their own sampling protocols. And this is my last slide. The classification scheme really has just core and then supplemental and exploratory sort of a graded approach that you can see here. And to summarize the way we've done this is we've taken a top-down approach to make it really easy for data entry. Is there tau, yes or no? If there is, then you go down in an iterative approach. What are the optional fields to permit novel sampling and other novel assessments so that we aren't constrained by specific pathological guidelines, although it's all based in our NIAA work up in NAC data structure. So we're doing the baseline stuff that we do for ADRC and the CTE consensus, and then hopefully this could be useful eventually as maybe a TBI supplement, just like we're thinking about VCID. So I'm going to stop. I know I probably went long. I apologize. Happy to take questions if there's time.
We have time for one quick question if someone wants to race to the mic. It looks like we have someone here.
Hi, Dirk. Great work. Henrik Zetterberg from University of Wisconsin and University of Gothenburg. I got curious about the blood samples that you also mentioned that you collect. Are those ante-mortem samples or post-mortem samples, and would they be available for validation of blood biomarkers against neuropathology? Ay. It was a good question. I can email you, Dirk. No, he doesn't hear.
I'm sorry. I just heard the last little bit that somebody could email me. If there was somebody asking a question, I didn't hear it.
Yeah, Dirk, do you hear me now?
I do hear you now.
Yes.
Sorry about that.
Henrik Zetterberg from University of Wisconsin and University of Gothenburg. Hi.
Hey.
Great work. I was thinking about the blood samples you mentioned. Are they collected ante-mortem or post-mortem, and would they be available for validation of blood biomarkers for neuropathology?
So it's only post-mortem at this point. We don't have any resources to meet these people before they die. But that's something we've discussed, is when we get a referral, how many of these people could be referred to our centers, et cetera. So it's all post-mortem, Henrik. So I don't know if that's valuable or not, probably not. But currently we're not doing any antemortem sampling in this pilot.
We are actually in Gothenburg studying biomarker stability post-mortem now in 1 day, 2 day, 3 days post-mortem. So perhaps they will be stable. It's not impossible that they might be. Good, thanks.
Well, let's talk, because we're taking that on everybody.
Nope. Can you hear me now? Hi, Dirk. This is
Yes.
from NIA.
Hi.
Yeah, the great work. I mean, really excited to see that something is coming out of this. And just was wondering if you could comment a little bit about the quality of the tissues that you're getting through this program, post-mortem times. Are you getting them early enough and you're able to do the full ADRC protocol? And I mean, if they are comparable to what you get through your regular programs?
Thanks, Eliezer. That's a great question. These are the same as we're getting through our program because we're consenting these people when they're alive. And so when they die, they have the same instructions as our clinical core participants for their families to contact us. So our average PMI for our center is 7 hours, and that's about what we're seeing for these folks as well. We have, I didn't present the numbers, but about 80% of our neurobiobank pilot here are rapid autopsies, meaning the postmortem interval is under 12 hours and gets the whole freezing program. They get our rapid MRI. They get everything that our clinical core people get. So from a tissue processing perspective, they're right there in the middle of the pipeline with everybody else. The difference is we don't know as much about them as obviously we do in our clinical core. But the benefits of this program are, A, we probably are getting the tissue, if not in better shape, in shape that is more appropriate for local uses to that community, and B, we are able to, maximize the infrastructure that we have to to collect and preserve and characterize those tissues as best as possible, again, for the local community. And so I think it's going to be really, I think if we can do this, I think these brains are going to be really valuable, because also we can use these donors for studies that don't need that longitudinal clinical data, which are really, really valuable, right? The people with all the biofluid biomarkers and the psychometric data, we want to keep those brains for studies that need that information.
Great, thank you.
Thank you so much, Dirk. So we need to move on to make sure we're on schedule. So without further ado, I'd like to introduce the first of 4 early stage investigators who are giving us brief talks. Ivy Nguyen is a faculty member at Mayo Clinic, and she is a participant in the Alzheimer's Disease Research Center as a neuropathologist, and she is going to be giving us a talk entitled Microglial Heterogeneity in Cognitively Resilient Individuals from the Mayo Clinic Study of Aging.
She's joining us virtually. We're just having 1 second to queue her up.
Recording in progress.
Hi, everyone. So good afternoon, everyone. My name is Ivy Nguyen, and I'm a neuropathologist and assistant professor at Mayo Clinic, Rochester. Thank you for the opportunity to present today, and I'm sorry I can't be there in person as I'm catching an international flight in just a couple hours. So today I'll go through one of my main projects, which is entitled Microglial Heterogeneity in Cognitively Resilient Individuals. While this is meant to be a data blitz, this talk may be more of a concept drip. So briefly, genome-wide association studies have identified over 30 AD genetic risk loci associated with innate immunity. Chief among these are ApoE and the E4 allele, as well as the TREM2 R47H variant, which both confer strong genetic risk for AD and are seen in microglia. During my postdoctoral fellow, this led us to ask the question, do AD risk genotypes associate with altered microglial phenotypes in human Alzheimer's brain? So using single nuclei RNA sequencing of human brain, we found a transcriptionally distinct microglial subpopulations that were enriched in cases with amyloid pathology, which is shown here in the far right T-SNP plot. So we term these population amyloid responsive microglia. So this is the ARM population here. To validate our transcriptomic findings and phenotype the microglia, we identified an IHC marker for the ARM population. CD163 was the highest differentially expressed gene for ARM, which we then used to stain Alzheimer's brain tissue. CD163 stained clustered and amoeboid microglia that were tightly associated with beta amyloid plaques, shown below, and the beta amyloid plaques are highlighted as the pink plaques. So armed with this marker, we stained an expanded autopsy cohort of Alzheimer brains with an admixture of ApoE and TREM2 genotypes. So this dot plot shows the CD163 to amyloid ratio taken from each case graphed as a function of the TREM2 or the ApoE genotype. The cases in blue are from the TREM2 wild type setting with various ApoE4 alleles. And as you can see, there's a stepwise decline in these cases with an increasing ApoE4 allele. A similar stepwise decline was seen in the TREM2 R47H setting, which is shown in red. So overall, the ARM in this case were significantly reduced in the setting of 2 of the highest AD genetic risk variants, ApoE4 and TREM2 R47H, underscoring how genetic risk influences microglial activation states. Now I'd like to switch gears just a little bit. As we know, AD is histologically characterized by beta amyloid plaques and neurofibrillary tau tangles that progress in a spatial temporally distinct fashion. With recent advances in neuroimaging and fluid biomarkers, it now allows us to study AD progression in vivo. So shown here on the right is a biomarker abnormality graphed as a function of clinical disease stage. In this model, beta amyloid is shown to accumulate even in cognitively normal patients, while tau accumulation occurs further along and may be more closely associated to neuronal injury and cognitive decline. And this is the premise of the ATN score, which was first described by Cliff Jack's lab. And I also think I wouldn't be a true Mayonite if I didn't include this graph. So I introduced the ATN framework in the last slide because it really changed my perspective on the relationship between neuropathology and cognition. When I started here at Mayo, I was introduced to this concept of cognitive resilience, the discrepancy observed in individuals with AD pathology and near normal cognition. So generally in the setting of significant amyloid or tau, and perhaps at some critical threshold, patients eventually demonstrate cognitive decline. This, of course, is influenced by genetics, education, age, sex, etc., which may directly affect brain structure, metabolism, functional network dynamics, and neuronal synaptic health. Interestingly, in several cohort and population-based studies, a minor subset of participants showed AD pathology without cognitive decline. This discrepancy between cognition and neuropath forms the basis of cognitive resilience. A recent GWAS conducted by Ramanant et al. Uncovered a novel association with cognitive resilience on chromosome 8 at the MTMR7 or C0.7 locus. Post hoc analyses confirmed this association was specific to the setting of elevated amyloid burden and not explained by differences in tau deposition or cerebrovascular disease. Ultimately, their take-home point was that genetic heterogeneity is one of the factors that may explain differential cognitive resilience to brain amyloidosis. So my driving question is, well, what about neuroinflammation, and what about neuroinflammation and resilience? In my prior project, I had seen that AD genetic risk variants significantly reduced the ARM population, again underscoring how genetics influences microglial activation states. So could ARM perhaps be protective? And if so, are they seen in resilient individuals? Also, what is their relationship to antemortem markers? So my current project focuses on defining the settings of individuals with significant amyloid or tau. The setting is ADNC neuropathology, as I am a neuropathologist. But we also include neuroimaging and fluid biomarkers data. We are going to collect the ApoE and TREM2 genetic data, as well as demographic variables such as education, age, sex, and evaluate ARM measurements and microglial morphology as a readout of microglial heterogeneity. By pairing this with antemortem biomarkers, we will hopefully identify various risk factors and potential predictors of the ARM tissue expression, shown here in the lower right-hand graphic. So an example of this is from our MCSA cohort. So here's an example of an 88-year-old male decedent. He's homozygous for ApoE44 with numerous lifestyle AD risk factors who remained cognitively intact prior to death. His MMSE was on average 28 out of 30 and was relatively stable, and his global cognition was also stable prior to death. There were no clinical concerns for cognitive impairment, and on multiple images, there was cortical thickness that remained normal, and prior to death, it showed very mild generalized atrophy. The participant reported high cognitive, physical activities, and education occupation scores well within the upper quartile for the 70+ population. So the neuropathologic findings were notable for high Alzheimer's disease neuropathologic change with an A score of A3, B3, C3. There was also severe cerebral amyloid angiopathy and multiple microinfarcts. When we evaluated the ARM population, the CD163 positive ARM were observed in the neocortices and the perivascular spaces shown here on lower power in the leftmost panel. The higher power image shows the clustered and amoeboid morphology appearing ARM. We then calculated the ARM to amyloid ratio, which was 0.26, and it was in the 90th percentile compared to the ApoE44 sex and age match controls provided from the University of Pennsylvania. So this case report from the MCSA underscores the potential interplay between microglia and resilience. And the driving hypothesis here is that the higher ARM may be seen in resilient individuals compared to age, sex, and ApoE genotype matched controls. So to test this hypothesis, we'll leverage the antemortem clinical data and integrate IHC staining of ARM and beta amyloid of postmortem tissue. The dot plot on the right highlights the global cognitive score as a function of age in the MCSA cohorts. And the dots in red represent individuals who scored in the upper quartile for their global Z-score compared to the same decade individuals. And from the cognitively intact group, the cognitively resilient individuals we defined as those with intermediate to high ADNC, determined by postmortem analyses. And comparisons will be made between the resilient group, those with low ADNC, and controls who are cognitively impaired, but matched for age, sex, and ApoE genotype, along with level of pathology. In the upper quartile so far, we've identified 118 participants who meet that 5-year cutoff. And this is preliminary data, of course, but out of the 118 cases, we were able to characterize about 103 of these cases shown here. ADNC was found in 97 of the participants, a third of which had the not likelihood, another third had the low likelihood, and a third had the intermediate to high ADNC likelihood. And on the right is a subset graphic demonstrating the ranges of the Thal and Brock stages in these cases. So in terms of evaluating microglial heterogeneity, I plan on employing two strategies, one of which will include the evaluation of ARM using % positive area. And the second strategy is in collaboration with the IFORIA group, which will employ an AI-based machine learning algorithm to recognize various microglial morphologies. As aforementioned, we can now study advances in AD using in vivo biomarkers. But ultimately, my goal is to integrate postmortem tissue pathology with antemortem neuroimaging and fluid biomarkers to better understand the neuroimmune response in aging and resilient individuals. Another exploratory, or what I would call a side hustle aim, is to evaluate the potential protective effects of ARM using 2-photon in vivo imaging. So the take-home points are that there is a level of microglial heterogeneity in Alzheimer's disease. In my postdoctoral fellowship project, we looked at the transcriptomic and activation states of these subpopulations, as well as characterizing an amyloid responsive microglia subpopulation that was reduced in the settings of genetic risk. I also tried to lay the framework for the cognitive resilience, or the capacity to cope with neuropathology. And some future directions include evaluating this heterogeneity in these resilient populations and how we are going to define resilience. Ultimately, I'd like to integrate these multidisciplinary approaches to demonstrate that, microglia, time and space do matter. In some ways, they're very much like time lords. So with that, I'll end with the acknowledgements, those from the Mayo Clinic, the ADRC Neuropath Core, especially Eleni, for helping me gather some of the preliminary data. I also want to say thanks to Dr. Prashanti Vermuri and Dr. Longjin Wu here, who have been great mentors. And also a special shout out to Dr. Addie Lee from UPenn, with whom I did my postdoc fellowship.
Fantastic. That was a wonderful talk. I hope we have time for questions, but we're going to move on to the next speaker, Dr. Michael Miller. He's a principal investigator and neuropathologist at Brigham and Women's Hospital at Harvard Medical School. He opened his independent laboratory in 2022. Focusing on mechanisms of somatic mutation and other aspects of single-cell biology in neurodegenerative disease. And he is joining us remotely to give us a talk entitled Somatic Mutation Profiling and Pathogenesis of Alzheimer's Disease by Single Neuron Genome Sequencing.
Yeah, and just a heads up about questions. We have time reserved from 3:05 to 3:15 for all of these Data Blitz presenters.
All right, thank you. Am I on? Great, thank you. So, hi everyone, good afternoon. I'm Mike Miller. I'm an assistant professor at Harvard Medical School and neuropathologist at Brigham and Women's Hospital. And I'm excited to tell you all about our work from the perspective of somatic mutations in Alzheimer's disease. This is a project that we did in collaboration with the Massachusetts Alzheimer's Disease Research Center, especially Matt Frosch and Brad Hyman and Derek Oakley there. So somatic mutations are well known in cancer and in recent years have been identified in non-neoplastic settings, especially in proliferative tissues like blood and epithelial organs, and also have been observed in post-mitotic cells in non-proliferating populations, including neurons, where there are hundreds of somatic single nucleotide variants in the brain. And these variants are very interesting for a variety of reasons. One is that they can be used to trace the lineage of cells in the brain, for example, identifying developmental relationships between neurons and between potentially other populations. Also, work has been done in the lab of my postdoctoral mentor, Chris Walsh, to look at somatic mutations in aging in neurons, finding that there's an increase, something like 20 somatic mutations. Each of our neurons gains that many for each year that we're alive. And so seeing these discoveries, I was interested to take a look at age-related neurodegenerative diseases like Alzheimer's disease to examine the potential biology of somatic mutations there. So in Alzheimer's disease, neuropathologically, of course, we think of it in terms of amyloid plaques, like some of the work that Ivy just talked about, and neurofibrillary tangles intracellularly, including tau. And with these pathologic observations and really decades of important mechanistic work, we have a general paradigm for how we think the pathogenesis of Alzheimer's occurs, which may be initiated by A beta misfolding into oligomers and also involve microglial activation, generation of reactive oxygen species, tau misfolding into tangles, and ultimately culminating in the death of cells. But we don't really have a great understanding for how this actual death of cells occurs. One hypothesis has been that perhaps there's genomic damage in the form of somatic mutations. And so we were interested in testing that hypothesis. So to do this, we use single-cell whole genome sequencing using postmortem tissue, human tissue. And this project, again, is a collaboration with the Massachusetts Alzheimer's Disease Research Center. So in our approach, we dissected brain tissue from specific regions, here focusing on advanced Alzheimer's disease, Braak stage 5 and 6 cases, and looking at the hippocampus, at the CA1 region, and at the prefrontal cortex. And we isolated individual nuclei from these cases, specifically the nuclei of neurons, using NuN as a marker in fluorescence activated nuclear sorting. We then amplified the genomes of those single neurons about 1 million-fold to enable us to do single-cell genome sequencing. And then from there, we did a variety of informatic analyses to identify the burden of mutations, as well as to look at the signatures, which I'll talk about in a moment, and various patterns of biology that might be related to the mutations. So here I'll show a few data plots that are in the same format, and we're plotting the number of somatic mutations, specifically single nucleotide variants on the y-axis, against, on the x-axis, the age of the patient at the time they died. And so here we are looking at controls with blue data points. Each represents a single individual neuron and a trend line in blue. And so blue is normal aging, and we can see that there's an increase with aging, as has been previously reported, and has a certain slope that we can characterize. It's something like 15 or 20 mutations per year that we're seeing. And in Alzheimer's disease, represented by the red data points and the red trend line, there's an increase in the number of somatic single nucleotide variants. We can see this both in our linear regression look against age, as well as in a direct comparison between Alzheimer's disease and the aged control cohort group. And these mutations, you might wonder, are they all present in APP or an Alzheimer's disease-related gene? They appear to be spread quite broadly across the genome. There's a fairly even distribution of them. We're seeing them on every chromosome, and so not just falling in particular genes, although there are certain enrichment patterns. One of the things that we did to try to understand what's causing these mutations and what they can teach us about Alzheimer's disease is to use mutational signature analysis. This is a method that attempts to use the pattern of which bases are changing, the nucleotide changes, to deduce the upstream causes of those mutations. So in cancer, where this analysis was pioneered, one can look at different cancers, for example, melanoma and lung carcinoma, and see these specific differences where melanomas are dominated in their major signature by C to T mutations and lung carcinomas by C to A mutations. And interestingly, if you were to take cultured cells, which this group did, and put on them a particular carcinogen that relates to those cancers, you see a very similar pattern. So in UV light, we see in cells a very similar set of mutations as in melanoma. And in benzoate-pyrene, one of the carcinogens in tobacco smoke, we see a very similar pattern as we see in lung carcinoma. And so in this way, we think that we can deduce the upstream causes of those mutations and mechanisms occurring in cells from the pattern of those mutations. So in Alzheimer's disease, we characterize two specific signatures in the somatic mutations in neurons, signature A and C, that have quite different patterns. A is age-associated, so it goes up over time with age, and it's somewhat similar between disease in red and controls in blue. There's a slight increase in Alzheimer's disease, but it's not statistically significant in most of the ways that we've looked at this. And in contrast, signature C which is comprised of a whole different set of base changes, we see an increase in Alzheimer's disease much more significantly compared to age. And so we're really interested in understanding what is this set of mutations that's going up in Alzheimer's disease that's selectively enriched in signature C. One group of them are C to A mutations, which have been previously associated with oxidative damage. So we did some work to look specifically at neurons and the amount of DNA oxidation using an immunofluorescence assay that uses 8-oxoguanine, the most commonly represented oxidized nucleotide, as a surrogate for oxidation in a cell. And what we see is that in Alzheimer's disease, there are more oxidized nucleotides than in controls. So with these two different methods, we're seeing results that seem to align and indicate that oxidative damage is at least one of the contributors to somatic mutations in Alzheimer's disease neurons. We also did an analysis where we looked at the density of mutations according to gene expression level. So in genes that are highly expressed in the brain, we see more somatic mutations. In genes that are less expressed, we see fewer mutations. And interestingly, while this single pattern emerges when we look at all the mutations, if we break it down by the two signatures, again, by the types of base changes, we see there's actually significant differences. Signature A, the age-associated one, is really responsible for this increase, this enrichment in mutations in expressed genes, whereas signature C shows a flat and if anything, a little bit of a negative relationship there. And so, these seem to be indicating that transcription, that the expression of genes is playing a role. And in some other data that I don't have time to show today, we've found evidence that nucleotide excision repair, which is coupled to transcription, may be involved with this process. So we've talked a bit about what's happening upstream, what's causing these mutations. Let's think a little bit about what's happening downstream. We can model, based on the number of somatic single nucleotide variants that we observe, the likelihood of a so-called knockout neuron, a neuron with biallelic inactivation of a recessive gene or monoallelic inactivation of a dominant gene. And based on this model, we can predict the number of neurons, the proportion of neurons that would have knockout genes. And we see that there's a significant increase in Alzheimer's disease compared to the controls. So one potential downstream effect has to do with direct functional impact on genes. Another one may have an immunologic tie-in. So we find that there's an increase in non-synonymous mutations, mutations that change the amino acid and potentially lead to expression of a neoantigen by that neuron. And so a neoantigen could be presented on the MHC class 1 machinery to responsive immune cells, especially CD8 positive T cells. And there has been a report that there are increased clonal CD8 positive T cells in Alzheimer's disease. It's not clear what they're responding to. One possibility is that they're responding to somatic mutation-generated neoantigens. So, I've told you a little bit about our observation and potential upstream and downstream effects of the somatic mutations. Just try to pull this together in a model that we're considering to try to make sense of all this data. Alzheimer's disease may be initiated by A beta oligomers, leading to subsequent events, including tau misfolding into tangles, oxidative damage, and then that and other processes lead to damage to DNA. And that damage is often repaired, especially by nucleotide excision repair, which is transcriptionally related. But under some circumstances, if the repair mechanisms are overwhelmed or if they perform misrepair, that can lead to somatic mutations and potentially to cell death. And we're actively interested in a whole range of questions about how these mechanisms really are interplaying regarding somatic mutations. So I'll close with that. I just want to thank all the many people involved with this project. Most of what I showed today was a collaboration with my postdoctoral mentor, Chris Walsh, and with computational biology colleagues, August Huang and Alice Lee, as well as with Mike Lodato at UMass. And many thanks to Brad Hyman, Matt Frosch, and Derek Oakley and others at the Massachusetts ADRC for their contributions to the project. And finally, just want to let everyone know, just a few months ago, I started my own independent lab at Brigham and Women's. And we have a small fledgling group, and we're really excited for more people to join us. So welcome talented scientists as we try to unravel the specific mechanisms of somatic mutation, how they relate to Alzheimer's disease and other neurodegenerative diseases. Feel free to look up more information on our lab site, and I think we'll take questions later as a group.
Thank you so much. We're going to move straight to our next speaker, and everyone else is in person. So next, I'd like to introduce Dr. Arlene Faustin. Dr. Faustin is a clinical assistant, a professor of anatomic pathology at NYU. She serves on both the clinical neuropathology and autopsy service. She's also been a neuropathology associate core leader since 2017. And she's going to give us a talk entitled Proteomic Studies on the Phosphorylated Tau Interactome in the Human Brain of Alzheimer's Disease. Thank you so much. It's a wonderful talk. And then last, we have Dr. Kurt Farrell, who's assistant professor of pathology, molecular and cell-based medicine at Mount Sinai. And his talk is going to be entitled Computer Vision and Machine Learning in Digital Neuropathology.
Thank you, Dr. Teich, for that lovely introduction, and thank you to the organizers of the conference for having me speak today. The title of my talk is Computer Vision and Machine Learning in Digital Neuropathology. So I'm going to start by giving a brief overview in machine learning and digital pathology, and then I'm going to go over a little bit of my current work, which uses supervised, semi-supervised, and unsupervised approaches to look at digital neuropathology. I'm just showing a little figure here from a publication that shows within pathology there's been an exponential increase in using machine learning and AI, and this can be applied to neuropathology to understand Alzheimer's diseases and related dementias. So what are the types of machine learning models? So artificial intelligence is a software that mimics models of human judgment. And then there's expert systems and machine learning, which we're focused on here, which learns patterns and relationships from data. And then there's certain algorithms that can be used to study our cases. They can be supervised and unsupervised. They include neural networks, regression methods, clustering models, things that we're vaguely familiar with to use, and they're not just a black box. But machine learning itself can be a little bit complicated. So most machine learning methods incorporate a central algorithm that processes each instance feature to yield an output for that instance, or what you see here in this schematic. The output may be a continuous variable in regression models, or a category and class label in classification models. The algorithm may implement a simple or complex equation, or it may represent multiple sequential network processing steps, as in a decision tree or a neural network. During learning, the output of the algorithm is progressively modified based on the data input. The learning process, referred to as training, does not modify the programming code or the algorithm in most forms of machine learning. Instead, the algorithm contains parameters, sometimes weights or bias values, and when we get those outputs, we can then apply them to other images that the model wasn't trained on. So this is a slide that shows a bunch of muffins and chihuahuas, and we can look at this slide and our brains can process it, and you can pick out what's a muffin and what's a chihuahua. But just until a few years ago, machine learning models couldn't really do this, but we have new graphical processing units and parallel computing clusters that now allow us to deploy models that can actually very easily identify these two things. And this is a good parallel because when you look at neuropathology, in particular a neurofibrillary tangle, it's not so easy to differentiate, especially for a machine learning algorithm. So back in 2019, we sought to make the first fully supervised machine learning model to just go ahead and pick out neurofibrillary tangles. And we used a SegNet architecture, and we had an expert pathologist annotate about 3,000 neurofibrillary tangles, and we trained this model, and then we took the weights from that model and deployed it on images that the model had not seen yet. And if you look at the bottom, the recall, the precision, the F1 score gives us kind of a good feeling that the model is in fact properly picking out just neurofibrillary tangles. So recently, we went to take this model on a larger cohort of individuals, those with primary age-related tauopathy. These are people that are tau positive but amyloid negative or just have low amounts of amyloid. the approximate number of cases and controls here, or people with cognitive impairment and not cognitive impairment, were 300 and 300. And what you can see is what you'd expect. The classifier went in, counted the tangles, and found in the hippocampus that in the entorhinal cortex in particular, there are significantly more tangles in those that were impaired versus not impaired, and then the combined hippocampus proper, we observed the same thing. And then we adjusted our model to correct for age, and it still holds true that more tangles meant that you were likely to be cognitively impaired, and this outperformed conventional approaches such as the BRAC staging system. In this same kind of study, we then looked at how the clustering of the tangles or the closeness to one another could potentially predict cognitive impairment. And what we found that if the tangles were more clustered or close to each other, it did in fact accurately predict whether the individual had cognitive impairment during their lifetime. And we think this could be interesting in other primary tauopathies such as chronic traumatic encephalopathy, where you see more focal lesions in those that are impaired. So in conclusion, AI-based counting of individual tangles across the medial temporal lobe was a strong predictor of cognitive impairment. Despite having some amyloid, our model still worked, and we also employed a novel graph theory of spatial clustering that also predicted cognitive impairment. Okay, so then we went ahead and we said, what if we did weekly supervised learning? And what if we did it on something that wasn't stained for pathology? So we took a hippocampus that was stained with LH&E. We then made a bunch of little tiles out of it, and we fed it into a convolutional neural network and told the network, some of these tiles, the people had cognitive impairment while they were alive, and some of the people did not. And it goes through and it runs the algorithm and it makes these training rates, and then I'm not going to go into all the math, but it produces these lovely heat maps that you see at the bottom that said what regions of that particular slide it was able to use to predict cognitive impairment or not. And what it focused on actually in this study was the white matter, and that was really interesting to us. So we took the tiles and observed them like any good neuropathologist would do. And what we found in the non-cognitively impaired group, the tiles were much more blue than the cognitively impaired group. And we thought maybe this was a proxy for myelinization. And so we then just quantified the amount of blue, and it was significantly different between the two. And to us, we felt was maybe indicative that there was something possibly going on to the white matter, and we're going to have further follow-up studies to figure that out. So again, despite noisy labels, because these people, our measures of cognitive impairment might have been done 3 to 5 years before they died, we still found that our semi-supervised trained model performed significantly above chance at predicting the presence or absence of cognitive impairment. And our interpretation studies on the macroatomic level showed that the attention tiles, there are differences in the myelin staining, and this could be suggestive of something that maybe we weren't looking at before. All right, so one more model to talk about, and this one is probably the most bizarre of all three, so you're going to have to bear with me on this. There's an article in the Times that came out maybe a year or two ago where we used machine learning and fed it different faces, and it made synthetic or fake images that looked very similar to what it trained on. And what it also produced is these latent space vectors that you could kind of play around with, and you could change things in the faces that it made, such as the orientation of the face, the color of the eyes, the hair length. So we thought this was really interesting, and we already had 3,000 neurofibrillary tangles that we had identified from the first study I spoke about, and we just went ahead and fed it into the model after we got it set up. And what it made was fake tangles. So you can see the training data. Those are actual neurofibrillary tangles from subjects. And in panel C, those tangles don't exist. The model made them. And I can go into the math of how it GAN works, but there's not time today, so just feel free to email me. So then we thought, what are we going to use with these fake tangles that we made? And we went back to an earlier paper published by Heiko Brack, and he described kind of the cytoskeletal sequence of what happens to a tangle as it evolves over time. You see some neurites retract back, soma size changes. And when we went back, because there's thousands of latent space vectors that we could play around with, we saw that when we modulated them, we did get some of these biologically relevant changes that you would see described years earlier by other neuropathologists. And working on a way to maybe use this to see maybe there's some sort of change that we're not noticing that the model can get us, and we can recapitulate it back onto the original tissue. At least that's the thought. I'm open to suggestions on how to continue this type of research because it's ongoing. So to conclude, GANs can be used to generate highly realistic synthetic microscopic pathology that accurately captures the breadth of biologic morphology. GAN latent space contains key morphological features that recapitulate the process of neurodegeneration and tangle evolution. And GAN based on supervised learning is a promising approach to understand histopathological staging of neurodegenerative disease. And with that, I'd like to particularly thank the Mount Sinai Alzheimer's Disease Center. They provided me REC funding that covered most of this work, so thank you, Mary Sano, for that. The NIH, which also has funded some of our work. The laboratory of Dr. John Crary, my colleagues at UT Southwestern, the computational unit at Mount Sinai as well. So with that, I will end right on time. Thank you.
Thank you so much. And unfortunately, we are running over, but we maybe have time for one question. If anyone has a question for any of the speakers. I have a question for Dr. Nguyen. For your amyloid-associated microglia that you found, did you, I mean, there have been other transcriptomic studies of microglia, and there's a variety of others, especially in mouse literature, groups of genes that have been associated with the transition to an AD phenotype, like the disease-associated group and others. Did you look to see if that was overrepresented or maybe even underrepresented in your microglial population?
Yeah, so great question, thank you. We did take a look at whether there was overlapping differentially expressed genes in the ARM subpopulation and the mouse literature. There really weren't in terms of the DAM paper and also the microglial neurodegenerative phenotype papers. And another actually reactivated response microglial subpopulation, there really weren't that many overlapping genes. I think the most prominent one was SPP1 or osteopontin, and maybe in a few subsets, ApoE. We included some of that data in the supplemental of our paper from 2020, so it is included there. And I think more recently, Marco Colonna came out with a really nice review of the different transcriptional states that were found in the human data and also the mouse data and whether they overlap and how they overlap.
Wonderful. Well, I have questions for all the other speakers, but unfortunately we have to move on. Thank you all for very great talks. Next, for the next hour, we're going to have two DEI-themed talks. And for the first speaker, I'd like to introduce Dr. Michael Williams. Dr. Williams is pursuing a neuropathology fellowship in the University of Alabama at Birmingham in the Department of Pathology. And he is founder and host of Diversify and Path, a community and podcast that focuses on the importance of investing in diversity in the field of pathology and laboratory medicine. In August of 21, Dr. Williams launched the Diversify and Path in hopes to grow a meaningful platform for diverse voices to be heard and learned in the field of pathology. And he has since released 40 episodes. And so, Dr. Williams, please take it away.
Great. Thank you all so much for allowing me to do doing a presentation on the podcast. And so, again, my name is Michael Williams. I am a neuropath fellow going to do forensics. Started this podcast last year, currently in season 3. So I'll just want to have a brief journey about starting a podcast. So I have no conflicts of interest or anything like that. So I want to start out with this first slide. So I'm a millennial and I talk in memes. And so for me, when I first seen this, I think this was during the COVID. This was during the height of the pandemic. And so I laughed a bit, but also cried a bit as well, like not crying tears, but actually like, oh no. Because we had a lot of stuff happen throughout this year. Besides COVID pandemic, it has unearthed, I guess, truths that for some people they were not aware of, but for people like me, it was kind of like, yeah, we've known this is happening for a while, but at the same time, we know that racism, for example, along with something I'm going to show on the next slide, are things that are factors that are that affect us. And so, in medicine, it seems like talking about other stuff besides medicine was kind of taboo. But I'm glad now that we're able to have conversations more about things that affect us as physicians and also affect our patients outside of just the normal, not normal, but things like cardiovascular disease, which is also equally important, but what's leading to that and what's causing that. And so this is, the next slide is going to be it's a trigger warning, so I'm sorry for those ahead of time, but I want to talk about the death of George Floyd. Another major thing, unfortunately, that happened when we see that. So being murdered, especially in Minneapolis, there were mass protests that took place in over 2,000 cities that was reported, especially in the United States and around the world. And so this next picture is showing some of the protests, or a picture of the protests. And looking at the signs that they are holding up does affect me. And so when I look back and when it happened, I don't think it was not surprising, but sad that there was not a discussion in the hospitals, or not necessarily in the hospital, but like amongst my, there wasn't a discussion about what happened. I had colleagues and friends who are people of color who we discussed and talked, and for us making sure that we were mentally okay. But outside of that, I still had to go to the hospital. I still had to be a resident and be in medicine. And one of the things that I found really striking was that how do we extricate, why is it that we have to extricate ourselves of who we are from the things that we practice in being medicine? I think we should empower ourselves, especially if we have ideas and thoughts to make things move forward. And so, throughout the pandemic, when we were isolating, one of the things I found that helped me was listening to podcasts. I really enjoyed hearing different thoughts and ideas, and I really said to myself, I wish there was one that talked about diversity in the field of pathology and laboratory medicine. And I don't remember the author, but somebody said, if you don't see something, create it. And so I created. So during APCP boards, and for those in the audience, anatomical clinical pathology boards, stressful time, but I was also learning how to be a podcaster as well too. So I guess it was pros and cons in one, but I love the fact that I was able to learn and do something else. And so I found a paper about podcasting, especially talk about racial justice, discourse reflection, and representation, especially within the graduate medical education. And so within it, it talks about the fact that podcasts can be used as a tool to achieve health equity, especially in the graduate medical education. It has the potential to combat racism in GME, and also can be used as a way to democratize and normalize conversations about race. And so with that, and with all the research I was doing in the back end, I created a podcast called Diversify and Path. And so I always start out the podcast saying thank you for listening. It's a podcast that explores how investing in diversity in laboratory medicine and pathology can lead to high return of investment, especially by learning from the diverse voices that's within our field. And so let me take a step back, because when I say that, it's interesting to me that I've had conversations with friends and colleagues who are people of color, who of course talk about microaggressions, and I say of course, but to me, I was like, these are conversations that we have. But again, these are not conversations that we have openly. These are conversations that I feel like at times were not normalized. And so I wanted to normalize that. I wanted to have a way for people to hear other people's stories, so that way, hopefully, at least there could be a one-on-one conversation for those who understand what it's like, or at least have different, conversations that may be difficult for some people, but also enlightening for others. And so I found a paper that talked about the effects of racial trauma on Black doctors or pharmacy students. And the reason why I brought this up is because it was very similar to what I was feeling during the death of George Floyd. Because of the fact that I'm hypervigilant, I'm scared, what's going to happen if I leave the house, leave my house and I get pulled over? What's going to happen? When they talk about racial trauma, they define it as a mental injury caused by encounters with racial bias, discrimination, and racism. And it's been seen and studied that it does lead to extensive negative effects of both mental and physical health. And so during that time, there was an increase in depression in populations of color throughout. And so I'll just go through this real quick, because I only have a little bit of time. So this paper had talked to pharmacy students, Black pharmacy students, surveyed them, and they found, based on their surveys and discussions, that they grouped the responses in 3 categories. So the first one is vicarious trauma, and the quotes is what one of the respondents had said. And so they said they didn't feel like George Floyd was any stranger to them. I felt very personal about it. Our dads are around his age, our cousins or our uncle. He was really a representation of our own families. And so seeing the death, unfortunately, had a way they felt that trauma as well too, because that could have been their father, that could have been someone that is close to them. Second thing is hypervigilance, a heightened sense of awareness, fear, and anxiety. And so one person had wrote that looking over their shoulder all the time, everywhere they felt that, even at work, they felt charged. I didn't write, in this also, they also talk about, there was one respondent who said they didn't exercise in normal neighborhoods they normally would because of the fact that they felt like they would have been stereotyped and they were afraid of any negative consequences. And the last one is fight or flight responses. So there was internal versus external manifestations of racial trauma. And so the internal one, one respondent said that the best way they can sum it up is to live through trauma because it happens, they still have to go to work and they're still required to live and make an income and feed their family. And so in a way, kind of bottling up what's happening. And then external, and so to me, I had reverberated with this because they write the fact that they took their approach of posting information about companies and they were conscious about it because they may not care whether that person dies or not. And so hopefully the next slide works. And I'm going to share some of the audiograms of some of the discussions I've had with people who've come on the podcast. Let me see. Nope. Can you not click on it? I think if you click on it, it should work. Okay. So.
And I don't know if my colleagues that go to medical school at HBCUs feel that same pressure because everyone is there together. I felt like there were a lot of times where my classmates were looking at me. Some of my non-Black classmates were looking at me like, oh, well, he only got in because, they had to meet a diversity quota or something like that. And I just don't know if that exists in other spaces. So I was constantly having to think about people looking at me, thinking that I didn't belong there. Imposter syndrome times 100 in terms of identity and race, not just in terms of academic and intellect. So that was another thing that I sort of was thinking about.
So this is Dr. Keenan Smith. He's at University of Pennsylvania. He's an APCP resident, and we discuss about being Black in medicine, what happens when we go to medical school. And I wanted to share, there were, this is, I guess, just the way it is, but there were people in medical school who have never really encountered a person of color before. So for me, I was like an ambassador. And that puts extra pressure on me, and because what kind of underlying biases based on TV do they get? And it's nothing against them, it's just the way that it ended up in medical school. So my second guest, the next guest is, Dr. Etta Lee, and we had a discussion about at least diversity in research. Can this go? There we go. Can you guys hear it? No.
No, any question you ask, the molecular genetics of X tumor or Y disease is not based on diverse cohorts, right? So in my field in neurodegenerative disease, a lot of what we understand that causes dementia is likely not exactly the same as what causes dementia in underprivileged groups or Black communities or Latino communities. And that's just getting started, right? Where it takes a certain amount of effort to go out and find these communities and enroll them into research, right? Because there's obviously a lot of hesitancy from people of color in trusting the medical establishment because we truthfully don't have the best track record. But as a pathologist, I think there's a lot of opportunity as long as you kind of keep your eyes open and if that's an interest, I think there's a lot of
Okay. And then the final one is Dennis Strang. He's a pathologist assistant, and he talks about allyship. Do I go forward with this? Nope. Can you press play again, please?
Being white, there was no possible way that I could understand these things. Because it's nothing, the things that you told me about your experience, in medical school, I'd never, obviously I'd never been to medical school, but even in college or whatever, I didn't have to experience any of those things. Nobody ever said to me, you don't belong here because you're white. you know, or you shouldn't, you're not good enough to be in this program because you're white. Like, that doesn't happen. You know, so I can't, you know, I would hear about these things and I couldn't understand them. So I felt like I didn't have anything to contribute to that conversation because it never happened to me and I have no idea what it's like. So I wouldn't, and I found that, and I found that, okay, instead of trying to go out there and say what I think.
And so he ended up saying he would just listen to what people would say. So I know in the interest of time, I will go through these next couple of slides so a little relatively faster. So sexual and gender minorities, umbrella terms for those sexual LGBTQ plus or gender minorities, those who does not identify cisgender. And so there were papers that looked at sexual and gender minority healthcare disparities, social barriers to care and strategies to bridge the gap. And what it said is the fact that there are health disparities in gender and sexual minorities, especially substance abuse, mental health disorders, unfortunately suicide attempts, and increase in sexual transmitted infections. And the fact that, especially for the transgender and non-binary patients, relating to access for medical service for gender affirmation therapy. The Human Rights Campaign actually talked about in the trans population, and so trans people live in poverty at elevated rates, especially for the intersection of trans people of color. There's stigma, harassment, discrimination. There's violence, unfortunately, against the trans people. And then there's also, at times, lack of health care coverage. And so I just wanted to show, this is Dr. Alcino Gama. He's actually at Northwestern University. He identifies as gay, Latino, and we have a discussion about that.
It happens everywhere. And if a program wants to embrace diversity, I think that they need to do that. And I see myself contributing to the gay Latin perspective of the diversity community.
Yes, that's me.
In the future, yes. I'm here for less than 3 months now, but I think that this is the flag that I am willing to carry, and I think that I can bring my perspective to the program and make us more diverse in that regard.
Okay, and then Dr. Dan Milner, and we have a discussion as well too.
I think, that's one of the fascinating things about Felix is that, Felix was a black gay man from Mississippi, right? And I was a white gay man from Alabama. So the only difference between us from a historical point of view is that he was black and I was white. But the level of respect that he had in the department was very high, very high. And I think it was a combination of the fact that he was so brilliant, he was such a good pathologist, he was a thyroid and cytopathology guy, just really, really good, fantastic teacher. And the department saw him for that, right?
And so we have a discussion about the intersection of race and sexuality, especially in the field of medicine. Unfortunately, the colleague he was talking about, Dr. Felix, had passed away. But he brought that up in terms of memory and having a correlation between being two Southern gay men, one black, one white, but also their experiences when they were working together.
I think,
Oh, okay. The concept of epidemiology of disability, I wanted to not end it, but also cover this aspect as well too, because I think it's one thing I'm sure that we all think about when we are looking at a DEI perspective. And so this is Dr. Ryan Clark, and he talks about his experiences on the wards after he was disabled.
Because you shouldn't be a doctor if you can't do those on-calls without being in agony after them.
Right.
So it was a very different experience. Didn't have all of those kind of experiences as a non-disabled student, and it was very eye-opening. I'll never forget going into my first meeting and having a meeting with the head of the year, who said to me, "You'll never become a doctor now because you're going to be too traumatized. You'll walk onto the wards and you just won't be able to cope." worst thing you can ever do to me is tell me that I won't be able to do something, because that will just make me do it. I'm very, very stubborn.
Right, yeah.
So I continued to excel after that because no one would tell me I couldn't be a doctor.
Because so the last question I always ask my guests are, how would you diversify in pathology? Now this could be broad because I know here we're talking about diversity, especially with ADRC, but I say pathology and laboratory medicine in general. How do we go about doing that? And so this next clip is Dr. Nicole Jackson. She was my first episode in season 1, and we have a short discussion about that.
A positive from it. And so there was a question posed very similarly to your question by the pathologist, and they wanted an interactive conversation. What I found so shocking was the response of at least a few people that did not realize how underrepresented minorities are. And it just never registered to them until people started posting articles such as Marissa White's article that reviews, the past few decades of how underrepresented minorities have been represented specifically in pathology. Short answer, we are equally underrepresented, if not more so than we were decades ago, despite all these diversity efforts. And people's responses were basically like, wow, I had no idea. Whereas for me or you or someone who's represented, we see this every day because you're usually the only face that looks remotely like you, and I think it just doesn't
and then the final is Dr. Joy Carter. She was the season finale for season 2. She is one of I believe, the first African-American female chief medical examiner. And she talks about her discussion. She discusses about her path into pathology and forensics, and also her voice for advocacy.
Like, okay, there's no one else that looks like me. But I'm at the table. I'm no longer on the stool. I'm sitting at the chair. And I said to myself, you're going to know that I'm here. And I said to them, I might be the first, but I guarantee you, I will not be the last. And that was my mission from that point on, to mentor and to expose the field of forensic pathology so that those people who would, if they see me speaking on CNN, they see me in a magazine article, they're going to hear my philosophy. They're going to know this is a field that we need so many more people of color, all colors. We need diversity. We need compassion. We need knowledge. You've got to get the knowledge.
So I just wanted to say thank you all for listening to a couple of the audiograms for the episodes. This comes out every Thursday with episodes that are about 45 minutes, sometimes an hour long. Thank you to the audience, of course. I also wanted to thank those who came on as guests. And so these are those from the first 10 episodes. And so I approached them individually on Twitter and I said, I want to start a podcast. I had no idea how to do a podcast, but they said yes. And so I was really happy that they came on and they allowed me to share their stories with those who are willing to listen. And then it just continues to grow on. So I've had multiple guests for season 1, season 2, and then season 3 is currently going on right now. And of course, I always think back about where I came from, where I did my training. This is my old residency at SUNY Upstate. I take a lot of selfies, by the way. And so these are a couple of people who supported me back when I switched residencies from surgery to pathology. And then these are those at UAB, basically just showing them the love that they've been giving me so far. So thank you again. This is me. And this is my QRS code, Diversify and Path. It's on most to all podcast platforms, and so you can download it, listen to episodes, and hopefully you get to hear somebody's experience. Thank you.
Hi, we have some time for questions. I have a question. I can lead things off. Thank you so much. That was fantastic. One thing that you said that struck me was the idea of visibility. So, being able to see, people from diverse backgrounds in leadership positions in pathology is important. Yeah. Your podcast is fantastic, but there's no visual component. So I was wondering, have you thought about maybe video recording this or putting it, so that people can actually see what's happening in addition to hearing it?
So the recording, I actually do a video with those for the guests. And I am experimenting with video editing. When people come on, I tell them it's not going to go out because they might not have their makeup did or anything. So they want to make sure they look good. So, and I get it. I'm like, I get it. I want to look good too. So, so yes, I would be experimenting now in the future.
Okay, well, thank you so much for, sorry, was there another?
Oh, I was just going to say, don't be shy, grab the mic. We got a couple of minutes.
We talked about this, but for everybody else, what resources does it take to do this? Like, you're investing in this, it costs money, like, what does this require?
Yes, so this is definitely an endeavor that is not free, and so if I can, I guess, kind of put everything together, some place to host a podcasting, so the episodes are placed on one website, which I have to pay monthly, the autograms that I make as well for the marketing, that is also something that I pay out of pocket for, getting a microphone. The editing so far has been free, so I downloaded a free software that does WAV files, so that's been free for editing. Along with, I wish I could have my sheet right now, along with other stuff that I use for marketing for the podcast, so it does cost money. So, but yeah, so there is a lot of resources. The microphones that I use, the headphones that I use as well too. Of course, I'm going for best value, like in terms of what I can afford. And so through extensive research, I found those at least. But so yeah, that goes on my budget every month about the expenses already for podcasts. I love doing it, so that's it. But yeah.
All right, well, thank you again. This is a fantastic talk.
Thank you. Sorry, I'm going to use it. Yes, so if there's any sort of grant or something for podcasting, let me know.
There should be. There may be. I don't know. But at any rate, so I'll introduce our last speaker, Dr. Amy Kine. Dr. Kine is the inaugural Associate Dean for Social Health Sciences and Programs at the University of Wisconsin School of Medicine and Public Health. Her leadership roles include serving as Executive Director of the Wisconsin Partnership Program Grant-Making Endowment, Director of the UW Center for Health Disparities Research, and leader of the CARE Research Core of the Wisconsin Alzheimer's Disease Research Center. And then she also does research in a variety of topics related to the theme of this meeting, and she's going to give us a talk entitled The Neighborhood Study, Linking the Disparities Aligned Exposome to Brain Tissue. Thank you so much. We actually are immediately transitioning to cross-core collaborative session number 1, and at 4:15, Dr. Amy Kine is going to be introducing that session. So before she introduces the next session, I would like to thank her for a wonderful talk and close the Neuropathology Core Session.